Okay, welcome back. We're going to finish up Nature via Nurture single gene effects by looking at a couple more examples. So I'm going to move to my PowerPoint, and this time I'm going to pop it out so that it's nice and big. We've looked at a couple of examples of how a single gene may contribute to behavior. Here are two more examples. One of the gene here is the ASPM gene and microcephaly. Microcephaly isn't in itself a behavior, but by the way, this gene is mentioned in our Agile Gene book at the end of chapter 1, pages, somewhere in the pages of 35, 36, 37, if you will. And so, first of all, I better explain microcephaly. Microcephaly is a condition in which the brain is smaller than it should be, and particularly the cerebral cortex, that outer layer of the brain involved in so many of our functions, right? In any case, here's a picture of a baby with a typical head size, with a big brain, and then a baby with microcephaly or severe microcephaly, right? Microcephaly can arise because of a whole suite of factors. Probably you've heard of the disease Zika, that's caused by or carried by mosquitoes. That contributes to one form of microcephaly. Okay, if you have microcephaly, individuals with it have intellectual disabilities, poor motor functions. Boy, have I not spelled either of those correctly. Poor speech, some seizures, some can have dwarfism, etc. Now, researchers noticed there are several families around the world in which microcephaly seems to be familial or inherited. And they realized that in many of these families, so here's a picture of a family tree and a couple of individuals in that family who have microcephaly. In these families, a single gene seemed to be implicated, and that gene is the ASPM gene, contributing to microcephaly and all of the behaviors associated with microcephaly. What is this gene and how does it work, right? This gene is called, here's a long name, abnormal spindle-like microcephaly associated gene. It's a huge gene. It's located on chromosome number 1. And what it does is it provides instructions for the making of a protein. Okay, that's what genes do, right? But this protein is involved in cell division. And in particular, this protein seems to be involved in cell division in cells early in embryonic development that are in the brain. So what it does is it regulates the protein, regulates the number of times that neuronal stem cells, so like these cells, these progenitor cells, if you will, that are going to divide, become the neurons of the brain. It regulates the number of times that these cells divide in a young brain. It regulates the development of neurons. And if you have a mutation in this gene, what that does is it impairs or prevents cell division in these cells, in the developing brain. And if you prevent or impair cell division, that's going to lead to fewer neurons, smaller brain, and all of the behaviors associated with that smaller brain. Okay, so having a mutation in this gene can contribute to the development of that smaller brain. And what researchers have found is there are a number of different mutations that are familial, found in families, that contribute to the genetic inheritance of microcephaly, if you will, because they're mutations in a single gene, this single gene, that contribute to the size of the brain. Wow, a single gene that contributes to a huge effect on brain size. Okay, that's pretty interesting. And researchers are really interested in it because my dog just hit my door really hard, made a big bang. I'm like, what did he do? What did she do? I don't know. In any case, what's interesting about this is back in our evolutionary history, we saw, relative to other hominids, a huge increase in the size, in our hominid ancestors, an increase in the size of hominid brains over time. Brains got larger and larger and larger for several million years. If a single gene could contribute to a huge change in brain size, wow, isn't that interesting? And that could contribute to a suite of behavioral changes and abilities, perhaps increases in our cognitive abilities because of a change in brain size, just because of a single gene or a mutation in a single gene. That's pretty interesting. It's really interesting because a lot of our hominid ancestors had smaller brains like this, okay? And chimpanzees do as well. So this is one of these genes in which researchers are kind of interested because chimps and humans are very, very different in behavior, but not that different in their genetic makeup. But if they're different in small ways, in single genes like this one, that could explain the massive differences in our brain development and in our behavior. So this is a really interesting gene that researchers are studying, and they're studying it in terms of the evolution of large brain size in humans. Okay, so that's just one example. Here's one more example before we go on how a gene can contribute to behavior. This gene is called the FOSB gene. It's found in mice, it's found in humans, it's found in lots and lots of animals, and there's a small discussion about this gene and its effects in mice in our book on Mother Nature, pages 146 through 151, so take a look at that. This is called a switch gene, or this is kind of a generic term for it. If you're a biologist, you might know what this means. What it does is it codes for a protein called a transcription factor. And transcription factors bind to and cause the expression of other genes. So a switch gene can kind of turn on lots of other genes. A switch gene produces a protein that can regulate the expression of other genes. Again, this is a situation in which a single gene then may have a big effect if it regulates the expression of lots of genes, right? Well, the FOSB gene, I think it's just called the FOSB gene, goes something like this. An environmental stimuli, or stimuli in the environment, turn on the FOSB gene, and the FOSB gene then makes the transcription factor that regulates a whole suite of neuronal responses, if you will. And what's interesting about this gene is that mice that are mutant at this gene, in other words, have a mutation at this gene, okay, they don't make any of the FOSB protein. These guys are trouble. So what they don't make any of this FOSB protein in particular their preoptic area of the hypothalamus. Okay, the hypothalamus is a part of the brain at the base of the brain there, and there are different areas. The hypothalamus is a big regulator, right? A part of the brain that's involved in regulating everything from appetite to addiction to sleep to libido or interest in sex, all sorts of things. And the mutant mice are missing, or mutants for this particular gene don't make the protein that's normally expressed in the preoptic area of the hypothalamus. But weirdly, these mice are just terrible at being mothers. They have a profound inability to nurture their young. And, all right? So researchers are really interested in this gene then, like, oh, here it is. We found the gene for nurturing. Again, we don't want to use that language. We found a gene that in some way influences nurturing behavior. Okay, let's say it that way. So researchers have done all sorts of studies on these mice. First of all, they realized that individuals who have this mutation, females who have this mutation, are terrible moms, all right? And so what we've got here are a couple of pictures, a couple of graphs that say the average surviving pups per pregnancies, okay? And there are a couple of different situations going on, but the kicker is these females that have the mutations here have two negatives, two negative versions of this gene, or two mutant versions of this gene. They can barely raise a single offspring, versus females who have who are either heterozygous, who are heterozygous for this gene, okay, who have one normal version of the gene. They're great, okay, they can raise their 8, 9, 10 pups all the time. And the same is true over here. If you're a mom who has this mutation, you are terrible. And here's two pictures showing how terrible you are. This is a normal mom. Normal mom picks up all of her pups, brings them under her, lets them, licks them, carries them to huddle together to keep them warm, and also allows them to nurse. And over here we've got the poor mom who's the mutant. And all of her little babies are just scattered around. She is not able to, or doesn't pick them up and carry them into a huddle, or it may take her like 50 times as long to do that. She doesn't nurse them. She doesn't keep them warm. She basically doesn't pay any attention to them. So the question is, what's going on? How does this gene how does a mutation in this gene contribute to this inability to nurture your young? What is going on? So researchers have asked this question, why are these mice so defective in nurturing? And they have looked at all sorts of reasons. They said, all right, well, maybe this gene somehow contributes to a change in hormones. So let's look at things like estrogen and progesterone and things like that, testosterone. Oh, moms seem pretty normal. What about like a structural problem in the hypothalamus and you're just unable to nurture because your brain structure didn't develop normally? Nope, that's not the problem. Okay, what about learning? Maybe these animals can learn. So they're pregnant the first time, they're terrible moms. But the second time or third time or fourth time, do they get better? No. What if they have a helper or a tutor, like another mom who's really good at being a parent, and they watch that other mom? Do they get better? No. Learning and experience don't overcome the deficiency. What about your sensory systems? Maybe these moms just lack the ability to smell their pups, and of course then they can't take care of them. Nope, that's not it. Maybe they're just not very bright moms cognitively, and so they're just not going to be good parents either. Nope, that's not it either. Wow, lots of problems here. Or lots of normalness here, I should say. What's going on then with these FOSB mutant moms? Why can't they nurture? Well, the idea is, at least now, we're not exactly sure, is that, these early genes, this would be considered an early gene that has a cascading effect that regulates a lot of neuronal responses that are adaptive, okay? It goes something like this. There has to be an environmental component, some exposure to the pups, probably an olfactory exposure, maybe tactile. They see the pups, they smell the pups, they hear the pups, something like that. And that triggers FOSB expression. So you start to see this gene being expressed in parts of the hypothalamus. And that in some way facilitates the strengthening of neural circuitry that enhances nurturing behavior. Researchers have suggested that perhaps the FOSB protein somehow enhances responses to oxytocin in the brain. And oxytocin is a hormone that has been over and over shown to enhance pair bonding and bonding in animals, and so that would enhance nurturing behavior. Maybe the FOSB protein somehow enhances responses to oxytocin, and if you have the mutation, you don't get that enhanced response to oxytocin, you don't get any nurturing behavior. I'm not exactly sure of this mechanism. This is kind of like the black box somehow, right? But we do know that the presence of a mutation at this gene contributes in a big way to an inability to be a good mom in a mouse, at least, right? So here's another example of a single gene, this one is a regulatory gene, one that codes for a transcription factor that influences lots of other genes perhaps. A regulatory gene influences behavior. It's a cool one. Okay, so some conclusions here are that single genes do play important roles in behavior, including social behavior, including complex behaviors like nurturing, like mother, right? But behavior is way complex. It's going to be affected by many genes, gene-gene interactions, gene-environment interactions, and we haven't focused much on those yet, all right? So for this lecture, you can think about understanding chromosomal theory, and what genes do, looking at the differences between humans and chimps, and understanding some of the examples of the genes that we focused on. And that's it. So I will escape here, get out of here, and turn off my video. Thanks so much.